Welcome back to the Business of Biotech. I'm your host, Ben Comer, Chief Editor at Life Science Leader, and today we're in beautiful Miami, Florida for a series of episodes recorded on location at the offices of Catalyst Pharmaceuticals. For this series, we're speaking with some of the people operating companies regionally here in the Sunshine State, and I'm excited to speak with Daniel Teper, founder and CEO at NAYA Therapeutics, a company developing cancer therapies based on alpha-emitting radiopharmaceuticals and bifunctional antibodies, two very hot modalities in drug development today. Daniel has a diverse range of experiences in the life sciences industry, spanning commercial and operational functions, advertising and consulting, finance and company founding. NAYA Therapeutics is the latest company he's founded. He's founded several. And I'm pleased to have him on the show today to learn about NAYA Therapeutics, its founding in 2023, what key opportunities exist in radiopharmaceuticals and bifunctional antibodies, and why he set up his company in South Florida. Thank you so much for being here, Daniel.
Thank you, Ben.
I mentioned your diverse background of experiences just now. You've worked in most, if not all, of the key functional areas required for biotech success. Why not simply go deep in a single functional area like commercialization, for example?
Well, I think in order to be successful in biotech, you need to understand the translation from the science to the clinic to the patient. And this is something that I bring to my leadership, but that the entire leadership team of NAYA also brings.
Having a diverse background of experiences.
Having a diverse yet deep background in each of the functions and being able to integrate those different aspects. So, of course, science, particularly at early stages, trumps everything. You need to have good science and good technology. But at the end of the day, you want to develop products that move the needle for patients and for physicians. So you have to constantly benchmark what you're doing and what you're developing to the patient needs and the competitive environment.
Right, yeah. We at the Business of Biotech, we like to get to know our guests a little bit through learning about their background. And so I want to get to NAYA Therapeutics, but I wonder if you could talk about a couple of experiences you've had prior to founding NAYA that taught you something important maybe, or served as a stepping stone to founding NAYA in 2023.
I'd like to say that being an entrepreneur and even being a corporate leader is like competitive sports. So you need to train a lot, but you also need to play a lot, and you need to have victories and setbacks and learn from your setbacks so that you play better the next time.
Yeah, you've worked. I mentioned you've worked in advertising. You've worked in strategy and consulting. You've had a number of different roles. Is there a favorite that you've had? Leaving out company formation and company leadership. Is there a functional area that you particularly liked?
I like putting together companies and projects. So the ability that I've developed over the years of taking different technologies, different products, and making it into something that's marketable.
Yeah.
That's one of my passion. And if you look at how we created NAYA and previous companies, it essentially is matching the vision to what is the reality, which technology do you need, which cancer target are you going after, what disease you're prioritizing. When I was a strategy consultant, I did a lot of portfolio optimization for big pharma. And so which indication you move first, and which you move second, and which you put aside. And you don't have to make a decision once for all. You have to monitor, is your data good enough? If it's not good enough, it's better to stop the project. So constantly have a target product profile, which sometimes, scientists, in all due respect, they fall in love with the science.
Sure.
And they don't realize that, they have to match or exceed the target product profile, constantly.
On that company formation piece, I mentioned in the intro, you've formed multiple companies at this point. Is there a playbook now for you? A set number of things like and maybe we can talk about it in the context of NAYA. What was the catalyst that led to that founding? And, what are the first things you do when you decide, all right, I'm going to start up a new company?
Yeah, I think the playbook is always evolving, based on what you learn and the circumstances. Clearly, what I've learned over the years is that timing is a critical success factor. If you're too early with an idea or too late, you can't succeed. And I have to say, we'll get back to it, is that with NAYA, we have the perfect timing for what we're doing. And, some of it is luck, but the luck comes to those who are prepared.
We're in Miami. What are some of the reasons for headquartering, founding the company in this particular place?
Well, this is not my first time in Miami. I've been back to Miami, 5 years ago. But clearly, the Miami of today is completely different than the Miami where I lived, 15 years ago. It's a bubbling metropolis. It is actually a world metropolis that is competing with New York and San Francisco and a number of European and Asian cities. And that's why you see people coming. It's not just a gateway to South America, and South America is very important, but I think it's the gateway to the world, and people are realizing that. And now we're getting to a critical mass of people that are coming from all horizons geographically, professions. You have great physicians, you have great financiers, all in Miami. Right. And that's a recipe for success.
You talked about timing in the formation of NAYA, so I'm curious. Let's dig into that a little bit. Why was the time right in 2023 to found NAYA, and was it because of the assets that you want to develop? The product candidates. Was that what made the timing right, or was it something else? What came together and spurred you on to start the company then?
So I think I will cover it from two angles.
Okay.
The first one is when you're looking at the genesis of unicorn companies. Typically, those unicorn companies have two attributes: is that they scale at the right time, so not too early. They know that there's a market need. They know that they have a technology that works and that can match the market need. And that's true not only in biotech, that's true across industry. And they validated that technology and that science works. If you do it too late, if you procrastinate on the science and you want to have more data, you can also fail, because in the meantime, you have 10 and 100 companies competing. I think one of the examples is the PD-1 VEGF antibodies, which is a project that we considered. And at the time we considered it, we had the right technology internally. We were fourths or fifths, and we had a chance to differentiate. And then within months, there was 153 and probably more today competitors. So obviously we put it to the side.
Are we getting to that point with GLP-1s, do you think? Or is it become so crowded in that space? Everybody seems to want to have one. I'm just curious about your thought on that, on timing-wise.
So it's all in the nuances. And I'll talk about GLP-1, but also about our assets. Is if you're developing last generation GLP-1, for sure, you're not competitive. If you're developing an oral one and you have a technology that works, that's a different story. And I'm oversimplifying, but that's the big idea. The same thing is true for the assets at NAYA. Some of the investors are telling us, oh, it is competitive. And you say, what does it mean it's competitive? Is there still an unmet medical need? And if there's an unmet medical need, like there is in hepatocellular carcinoma, our lead indications, where over 75% of the patients are not responding to standard of care and or relapse. And if you have half a dozen products in development, but the different modalities and different targets, and that you can be, either first in class or best in class, or you have a realistic shot at getting there, then it's worth going.
Right. Let's talk about those assets at NAYA a little bit more. I guess first off, where did they come from? Where did you get them?
So the theory that I was giving you, we did actually put it into practice.
Okay.
So if we're talking about the bifunctional antibodies, we sourced the bifunctional technology scaffold from one of our founding scientists.
Oh, okay. So it's internal.
It's not totally internal. He did it, in an academic setting,
Right,
but
sure.
we basically bought his rights
Okay.
to the technology. We identified hepatocellular carcinoma and GPC3 as hepatocellular carcinoma is an unmet medical need. GPC3 as a great target. Actually, GPC3 is probably today one of the very best target for solid tumors. It's an oncofetal protein. It's expressed only on tumor cells, on liver tumor cells, and a few other cancers, but it's really on the tumor cells and not on the healthy tissues. So that's a great target, and it has also a pharmacological effect by itself. Now, the other component of creating the bifunctional antibody is what do we do to accelerate the killing of the cells? I mean, it's accelerate and sustain the killing. And immune cells can be redirected to kill the tumor cells, and you can do it with T cells, you can do it with NK cells. T cells have been more successful, but activating T cells also has significant side effects and is more difficult to use.
Right.
NK cells, I would say the first generation of NK cell engagers has been disappointing because it didn't have a sustained effect. So we looked at the biology, we identified NKp46 as an activator of NK cells, which is consistently expressed in the tumor microenvironment, including in solid tumors, and that does serial killing, that reverse the dysfunction of immune cells. I mean, all those nuances of the science, you marry them to what are you trying to achieve clinically, and what you achieve clinically determines what's the market potential for your product.
Right. What about the radionuclide that you're developing? Is that a separate asset, or are they working together in combination? How does that look?
It's a synergistic asset.
Okay.
So it's not the same asset.
Right.
And obviously, we have backgrounds in antibodies. And in order to deliver radionuclides, you need a vector, and antibodies, and particularly antibody fragments, are appropriate. The story that's interesting is that the scientists, our scientists that helped us with the bifunctional antibodies also worked in radiopharmaceuticals 25 years ago.
Wow.
And even took a product to phase 2, successfully to phase 2 clinical trials. So when he suggested to me that there are some new radionuclides that we should consider that will help democratize the field, I was all ears, and the team was all ears, and I would say within a few months we were able to create new products. And remember that we have a bifunctional antibody for HCC that targets GPC3. GPC3 is also a great target for radiopharmaceuticals. Actually, Bayer and Bristol Myers Squibb are developing GPC3 actinium-225 radiopharmaceuticals. They're in phase 1 development, and obviously there's a reason why they're investing in there. So we said, if we take our GPC3 antibody, we define the optimal construct, and for radiopharmaceutical, a fragment is a better construct because it aligns with the half-life of the radionuclide, and we have the optimal linker, and we have the optimal radionuclide, and the optimal radionuclide we believe is astatine-211. And why is it optimal? It is very clean. It's clean for manufacturing. It's clean at the clinical level.
What do you mean by clean?
You know, radioactivity obviously can be dangerous.
Sure.
You have radioactive waste.
Absolutely, yeah.
You have toxicity. You have to shield the units where you're treating the patients with most of the, I would say, legacy radionuclides. If it has too long of a half-life, obviously you have a risk of bone marrow toxicity or kidney toxicity. So astatine-211 has been around for more than 20 years. Astatine-211 could be the Goldilocks of radionuclides. It has a short half-life, 7.2 hours. It has a clean decay, which, with a single daughter, which means that you don't have all the problems in the waste, in the waste management. It requires much less protection than the other, radionuclide. And it's been proven preclinically and in early clinical trials to work just as well, very precisely killing the tumor and particularly killing the isolated cells. I think what we forget often is that it's not all about bulky tumors and surgery and chemotherapy and immunotherapy. It's also about residual disease and metastasis. If you take prostate cancer, which is not an indication that we're pursuing now, you can remove the prostate with surgery. But there's still, you have a risk of relapse.
Right.
And what's the risk of relapse is that you have isolated cells that are silent. And so that's where radiopharmaceuticals are very effective. And I'm sure you're familiar with Pluvicto from Novartis. So it's using a beta emitter, which is more difficult, more toxic. So the next generations are the alpha emitters,
right,
which are actinium, lead, and astatine, and we believe that astatine is going to be the winner. There's a room for all three, but astatine is going to be the winner in the next 10 years.
So astatine-211, you have that candidate. You have your NK bifunctional antibody, and you say they're synergistic, and you're developing them both for the same indication, but are they administered separately? Or in concert with each other?
Sure.
How does that, yeah.
So astatine is not a product. Astatine is a component of the radiopharmaceutical.
Okay, right.
So you have one product, which is GPC3 NKp46. It redirects NK cell to kill the tumor cells.
Okay.
It is positioned in non-responders to immunotherapy, and particularly to checkpoint inhibitors plus or minus a VEGF inhibitor like Avastin. That's a huge market. That's 75% of the patients that are treated with drug therapy. The second product is GPC3 linked to astatine-211. So that's a radiopharmaceutical.
I see.
Its use is slightly different. So first of all, it's very appropriate to use it in the half of the patients that are not getting drug therapy. They're getting surgery, ablation, TACE, and that what you need radiopharmaceuticals around those procedures, before or after, to eliminate the isolated cells and to minimize the risk of metastasis. Now, later in the treatment, either after checkpoint inhibitors or after the GPC3 NK engager, you can also do a short course of GPC3 astatine, just to make sure that you're proactively handling the risk of metastasis. And there's even, I want to mention, because I just read it yesterday, there's some early experiments of radiopharmaceuticals in liver metastasis. So you can get liver metastasis from breast cancer and other tumors. So that's more on the explorational side, but that's, again, a huge unmet medical need.
There's obviously a lot of excitement about radiopharmaceuticals. Generally, some really big deals have happened in that space. There's a lot of excitement about alpha emitters in particular, as you've mentioned. In the past, supply chain, isotope half-life have added a layer of complexity to supply chain, manufacturing, commercialization. What can you tell me about, I guess, NAYA's approach to sourcing astatine-211 and how you're scaling up manufacturing?
Sure. So until, I would say, the last few months, astatine-211 was restricted to just a few academic centers, 2 or 3 in the US, 2 or 3 in Europe. Japan was relatively more advanced in terms of the infrastructure because you needed to have the cyclotron, the radiochemistry lab, and the patient in the same location.
Okay.
So that's very restrictive.
Right.
And what changed this year, and that's where having all the stars align is extremely important, is that the Atlas C100 machine from Atlas Solutions in Sweden was released, and that machine allows for automated purification, conjugation, and production of therapeutic dose locally. So essentially, without getting into too much detail, the way our products are being manufactured and will be manufactured in the future is that you need a central or regional cyclotron where you're bombarding bismuth with alpha particle, and that gives you astatine powder on the target, and that target can travel easily and can be shipped to a local lab where you can do the GMP therapeutic dose. So that local lab can be in Miami, can be in New York, can be in San Francisco.
Okay.
Bismuth, by the way, is widely abundant.
Right.
And it's very inexpensive. That's a big contrast with what we're seeing with the other radioisotope that requires source of uranium or nuclear waste. So that's a big difference. So our vision is essentially that there will be 5 or 6 commercial regional cyclotrons, and we have a partnership with Ionetics, currently based in Michigan, but they have plans to expand. There are other companies also doing that. And then you will have multiple decentralized GMP micromanufacturing units, which are not capital intensive. And I would say for the US, it's probably between 10 and 20, depending on the coverage that you want.
Okay. That's excellent. I appreciate that. I want to ask about NAYA's current financial situation and runway, but really, I guess what I'm looking for is what the current investor situation is like for a company that's in the early stages developing some really novel products. What's your read on the investor situation right now, and do you think it's improving or not?
I think after the summer, the investors are looking for opportunities, and the reason they're looking for opportunities is because they see the constant appetite of pharma companies for novel biotech. I think it's up to 75% of the products are being sourced from biotech companies.
Yeah, right.
Now, they're being sourced earlier and earlier. This is something that I always like to illustrate. The 3 or 4 transactions in radiopharmaceuticals are phase 1, phase 2 at the latest, sometimes even preclinical.
A couple of those are billion-dollar transactions
They're
too.
multi-billion dollars.
Yeah.
So I like to say that some of the best transactions, whether it's bifunctional antibody, by the way, or radiopharmaceuticals, they're $1 to $4 billion, I would say, in phase 1, 2. And once you get into phase 2, 3, it's 5 to 10 billion. I mean, we just saw this week Genmab, which is not a big pharma, it's a large biotech, acquiring Merus, which is in phase 2-3 for $8 billion.
Yeah.
So obviously that's attractive for investors because investors today are looking for exits in the relatively short term. So I would say and we do the same. So we're saying if there's not an exit can be out licensing, it could be an M&A, it could be an IPO. So if you don't have an inflection point that justify an exit strategy within 2 years, then it's much less investable. So in that context, NAYA has a pipeline with a clinical inflection points that support an exit. And again, it doesn't mean selling necessarily the company. It could be just out licensing 1 or 2 products or doing an IPO within the next 2 years. So we're currently raising Series A of $75 million with institutional investors. This is, in the range of the transaction, the high quality transaction that have been announced in the last few months, and that covers actually the execution of the development up to clinical data.
So what is your, from a clinical perspective, what is the milestone that's your top priority for the company right now? What's next for the 2 assets that you're developing?
So we are starting to treat patients in the first half of 2026 for the bifunctional antibody, and in the second half for the GPC3 acetine radiopharm. Because of the nature of oncology, those trials are open, and you get data as you go. So we anticipate that in early 2027, we'll have initial data, actually potentially on both of them. And by the end of 2027, we'll have more comprehensive data. There are a number of transactions that with only 30 patients have sparked the interest of pharma companies. So we want to be funded sufficiently so that we can progress on our own. But obviously, we're open to partnering with pharma to accelerate. We're also quite agile. So I'll give you an example. We're considering doing the initial clinical trial for the acetine product in China.
Oh, interesting.
And the reason of China is that you can do investigator-initiated trials faster, that you can get an IND. HCC is extremely prevalent in China. So you can recruit 20, 30 patients within a few months, and you can get substantial data.
Now, that would be, you would have to anonymize that data to bring it back to the US, right? Or would you be doing that in an effort to commercialize in China eventually?
So eventually we want to commercialize in China, or whoever our partner is would commercialize in China, and it's a huge market. But I'm sure, everybody is talking about all the deals that are sourced from China.
Yeah.
And whether it's pharma companies or VCs, unfortunately for us, they're sourcing deals in China.
Right.
So the question is, as an American company, can we be smarter and take advantage of the infrastructure in China to get data sooner?
Yeah.
And we believe we can do that. We have a partnership that we're planning to announce shortly with a company that has capabilities for GMP manufacturing.
In China?
In China, of acetine products.
Wow.
Which actually, that's a company that was started by 2 very renowned academic scientists from the US. So you have more and more movements. The word is not isolated. So it's coming from the US to China, from China to the US. I think India in the future will also rise. So anyway, moving faster, being lean, not spending money for the sake of spending money, putting the standards very high in the execution, but making sure that every dollar that we invest in development matters and gets us closer to clinical data.
I want to ask a question for the listeners of the Business of Biotech, many of whom are at very early stage preclinical companies or even thinking of starting a company. What case would you make to them about setting up their company in Miami, and what is some advice you would give to maybe a first-time company leader?
I would definitely, and I am recommending entrepreneurs to move to Miami. It's a very unique environment. Some of the other ecosystems are very mature. They're very crowded. Miami is rising. It's not crowded. It's entrepreneurial. Everybody wants to work together. You have easily access to key opinion leaders. You have easily access to investors, including institutional investors and bankers. And it's like immigrants moving to a new territory and wanting to build something together. And so that's a unique environment that I don't think there's any other place in the U.S. that an entrepreneur can find that.
Final question, and I'm asking this of everyone that I'm speaking with while I'm down here in Miami. It's a hypothetical, which is, let's assume you find yourself in the predicted path of a Category 4 or Category 5 hurricane. Do you board everything up and hunker down, or do you pack up the car and get out?
Are you talking about personal or for the company?
Personal.
I've been through hurricanes in the past, being in Miami, and I would say stay here. There are risks everywhere in the world. There's risk in California. There's even risk in New York City that water will submerge Manhattan.
Right.
There's unfortunately risks of war. So I don't think there's more risk in Miami than any other place.
All right, we're 2 for 2 on stay in town and hunker down during the hurricane. Thank you so much for speaking with me, Daniel. We've been speaking with Daniel Teper, founder and CEO at NAYA Therapeutics. I'm Ben Comer, and you've just listened to the Business of Biotech. Find us and subscribe anywhere you listen to podcasts, and be sure to check out our new weekly videocasts of these conversations every Monday under the Business of Biotech tab at lifescienceleader.com. We'll see you next week, and thanks as always for listening.
Thank you, Ben.